Yeah, yeah.
Okay, anyways, good afternoon everyone for the post-lunch talks. And the first speaker is Nika, and she's going to talk about mesoscale correlations in soft active materials. Yeah.
So yes, thank you very much. So I would really like to thank Pinaki and Arnab for inviting me here. It's very nice to be here. It's my first time visiting India, and I'm very much enjoying it to finally come to this country. So I wanted to essentially pick up where Ludovic left us this morning and talk more about mesoscale correlations in soft active matter, actually broadening in their scope even further from what we've had so far. So to follow up on what you heard this morning, people have talked a lot about active turbulence, and they put a lot of different systems together, which we can debate if they should belong together or not. Say, for example, bacterial suspensions, cell monolayers, microtubule kinesin suspensions, active pneumatics. And if you look at this from a distance, yes, they all look, I don't know, swirly, decorrelated, chaotic, or something like this, as you can see here in the Class of Active Turbulence review article of Ricard Allaire and Joanny. Now, if you look specifically in things like epithelial cell layers, like the ones here from my student Kaya, you will see that you see swirly spatiotemporal correlation functions, much like those you might see in a sheared passive material, for example, or indeed in an active glass. However, these have been interpreted over the last years often as some sort of feature of an active pneumatic liquid crystal or different types of active liquid crystal. And so what I'd like to convince you of is that this particular phenomenon and another class of related ones are all examples of something much simpler. They're examples of the mesoscale correlations you get when you combine active driving, polar active driving, uncorrelated, together with elasticity. So to keep it very simple, and this may be really the only slide we really need from this talk. Yes, everything else is details, yes. So the idea is very simple. So take a dense active material crawling over some kind of substrate, equations of motion, for example, here this is for active Brownian particles. So you have some sort of self-propulsion, which has rotational diffusion, which then will map to a time-correlated active driving with a persistence time tau, which is 1 over the rotational diffusion constant. You could also do Ornstein-Uhlenbeck active driving. It doesn't matter as long as it's persistent in time. Then if you're dense, this all corresponds to an equation of motion, which is for the displacement field u dot is some sort of active driving plus some sort of stiffness, Laplacian u in one dimension, so elastic deformations. Now I can put this correlation into this dynamical equation and I can solve it. I will get a correlation length in my material because of this overdamped dissipative driving, which is always the same, psi square root of kappa tau over friction coefficient. And that's it. So it's just you push for a certain time, and over this you get a diffusive propagation of the displacement, and that gets you the square root of tau scaling, which we already saw this morning. That's really all that there is to it. So what I'd like you to convince you of, is that this shows up in all kinds of systems. So I'm not certain I'll get through all of this. I'll see as far as I get. So I'll show you something in one dimension about traveling colloidal strings, which recently came out with my Bristol collaborators. Then I'll do cell sheets, ABPs, Carroll ABPs, applications to actual cell sheets, and if there is time, fluctuating active stresses, where you actually see the opposite with mesoscopic correlation length. So here is an experimental system of the group of Paddy Royal, then at Bristol, which we looked at together with Zichen Shao, my student with Tani Royal, Tani Liverpool. So you have a induced charge electrophoresis system in a strong electrical field, and in a passive system, these things make strings. It's a well understood system. Now these ones, they're active, and they're self-propelling along the x direction in here. Then in part of the phase diagram, among many other things, they make a traveling string phase here of moving active strings. I think I have a video here. You can see them here. It's not a perfect experiment because of sedimentation issues, but you can see them here moving. So inspired by this, we said, let's make a simulation of the system, understand what's happening in the dynamics of these strings. So what we did is we took a standard model, so WSA repulsion, and then dipole-dipole interactions between particles with the dipole direction always frozen along Z because of the strong field, but then now with self-propulsion orthogonal to the direction of polarization, so these kind of interactions. And if you put these together, as I said, with active driving, which is just active Brownian in the plane, XY orthogonal to the direction of the magnetic field, what you get is very nicely string formation, as you can see. So you can see traveling strings, and they're interesting. They're moving. They're also bending. They're having some very interesting correlations along the length of this particular of the strings. So once you have this, you can vary, of course, your parameters. And what you will find is that if you vary the amount of rotational diffusion, and you can also sort of see how far away from FDT, but that turns out to be not all that interesting, you get mainly two regions of phases. You get collisional strings, which is the ones you just saw on the previous slide, so moving strings which collide with each other with a finite lifetime. And you get these strings which just sort of sit there and vibrate and don't do very much. And also you can see they actually don't seem to be correlated. They're rather stiff. So it turns out we can understand most of this by just looking at a modified Rouse model of this system. So you can rewrite this all saying, okay, my string is pretty much a straight polymer, and let me develop everything in Rouse modes of deviations from that. And so you have your standard Rouse dynamics with stiffnesses which are a bit variable because you have an effective interaction between particles. But you can compute it actually. And then you have driving only in the XY plane, which is effective active with the same sort of persistence time correlation, which I showed you already a couple of slides ago. So if you take this together and actually work quite hard on it, you can eventually compute your tangent-tangent correlation function for this. And the tangent-tangent correlation function has a correlation length here in the distance between monomers, which is again psi a times square root stiffness over dr times friction coefficient. So 1 over dr is again the persistence time, so square root of tau in the system as well. And as you can see, there's a very good match between theory and simulations here for different dr, so you can see the growing correlation length. There's more to be said about this experiment, but not about the correlation length. So I would like to now go from 1D to 2 dimensions with this material. Yeah, too fast, yeah.
Well, I mean, my eyes might be fooling me, but wasn't there some sort of crystalline organization of the polymers in the video that you showed?
Not that I know of, not for the,
okay,
so.
It's my eyes that's fooling me.
No, no, we're doing 2D sheets now where it's more interesting, but not for this one.
Okay.
Yeah.
And
so I would like to go now to 2 dimensions. So in 2 dimensions, so paradigmatically, So this is the video I showed you already. Here are some active Brownian particles playing there in the corner. And so how should we interpret them in this framework? So if I just make the simplest approximation that I can and say I'm going to pretend I have an elastic solid there sitting there, yeah, or at least something which is only arranging on the time scale of the alpha relaxation time, which I assume for a moment is long compared to the persistence time, yeah. So I start with that assumption. Then I can pretend that in fact I'm trapped in some sort of energy minimum here of my energy landscape, and whatever my active dynamics is doing, it's doing it within that minimum, and then I can do some linear response theory and hope that it's a good model for my system. So if I do this, if I take linear response and uncorrelated activity like before, I can write my equations of motion as displacements being active driving. Here this is the Hessian matrix times the displacement, so good old granular material style Hessian normal mode approach. So if I take this, I can solve this fully, and I can get the energy spectrum out of this, and what I do is I break equipartition formally in the systems, because there's different energies for different modes, because as you can see it depends on the mode eigenvalue lambda mu here, and in fact it goes down for larger modes. The thing you see here in front here, zeta V0 squared tau, that's nothing over 4, so this is nothing that the effective active temperature for Brownian active Brownian particles you would normally look at. So you can see it's equipartition plus explicit modification here. So that's maybe interesting, and you can compute everything in normal modes, but there is more insight to be gained if you look at this in continuum. Sorry, there. So if you look at this in continuum, you can rewrite this, as I said already, as a zeta, so displacement field, dotted, active driving, and so we've written this dynamical matrix. I could write this like with Laplacian shear and bulk moduli, doesn't matter, yes? So but the interesting thing is that in Fourier space, of course, everything decouples, and I can write the active driving here. I can compute this. It doesn't couple to anything spatial, which is why we can solve all of this. And what it does instead, it's a colored active noise. So you're driving this with something explicitly non-thermal, with something Lorentzian, with a width which depends on the persistence time tau. Now in continuum, of course, you can solve this. I decompose into longitudinal and transverse components. I go to the two eigenmodes of that matrix D of k. So I have a longitudinal stiffness B plus mu, bulk modulus shear modulus, and transverse just a shear modulus. And if I do some calculation on this, I can compute analytically the velocity correlation functions in these systems directly. And what you can see in there is that they show up directly with a very standard classical kind of set of length scales. So one is longitudinal, one is transverse. There's a prefactor which goes like V0 square, because that's just to be expected. And then these length scales, they go like the square root of persistence time, bulk plus shear modulus over friction coefficient, or just a shear modulus, mode square root of tau. So as I said, this was the first relatively simple calculation, but this has since been extended. So Caprini came up with something very similar roughly at the same time, and people have since extended it to the liquid state and beyond, and so including some of the people present here in the audience. So we checked this for active Brownian particles. So you see left is a glassy phase, and right is a liquid phase, and you can see the velocity correlation functions here. So I should say the dots are numerics, the solid lines are the normal modes predictions, and as you can see, you don't see them. They're essentially exactly on top of the dots except down here. And then the dashed lines is the continuum description which I showed you. That works very well except here at this point, but this is the peak from the structure factor. So this is the peak from sampling where particles are. So that's not expected to come out of a continuum description. What's more interesting is that in fact this works even at much larger driving. So in the liquid phase, you can see it here, so I use this as the effective temperature. The bottom 4 of these are liquids. The first 2 of these are solids. And as you can see, they're all exact. They work very, very well, except for the 2 largest driving amplitudes. And in fact, I challenge you to figure out which one of these is the solid and which one is the liquid. Any guesses? So it's the top one, which is actually liquid. or actually I'm not even sure anymore. So in any case, this works well, and it works better than it has to for a linear response calculation. Now, so we know, so what I would like to claim is that this is another type of active turbulence, as Ludovic already said this morning. And in fact, I wanted to connect to his talk here, that in fact you can do this better. For example, Jan Keta looked at this, and the scaling is modified in liquid phase. And then if you drive even further, you can do the turbulence spectra and so on. So in fact, this morning's talk, imagine it fits now here. So then let me go on with something different. So once you have this, in fact, once you have this method, you can do much more. Actually, you can have fun. You almost have a machinery, because what you can look at different types of driving which are connected, but for which the same method will actually work. So the one I wanted to mention is work with Amir Shi, who was a postdoc with Christian Weppe, and who did his, he worked with people here locally as well. So if I take chiral active Brownian particles, so there's active Brownian particles which look like normal ones, but in the orientation I have a constant driving amplitude, so they rotate with a constant frequency in addition to the rotational diffusion. So these have been looked at intermittently as a type of chiral active matter, though I should say immediately it's not one of those which is which really has wave propagation or torque, yeah? So it's not autoelastic or something like this. But because of this, it's of the same class of what I just told you, and we can compute things. So we found something very interesting there. We found 4 different phases. So what you see here, this is a kymograph. So time along the x direction, space along the y direction. And we saw phases which have space and time correlation, including oscillations in a time direction. So the most interesting of these phases here is at low rotational diffusion constant and low chiral amplitude. So we call it CMRO, chiral mesoscopic range order. And this actually has been looked at already by the group of Lisbeth Janssen. They looked at the glass transition in these systems and found some rather odd type of effects in the melting and so on, which we think are actually related to the properties that we saw in them. So then Amir sat down, got maybe 2 coffees in a row, and then computed all of the correlation functions for this. And so in fact, we can solve everything. So you can do the same exact calculation as before. So you start with your own tangential correlation function. So it's an exponential decay, but it's modified by an oscillation because of the chiral amplitude, yeah? And because of this, you can again compute the energy spectra, and you again break equipartition, but you break it in a slightly more interesting way. So you break it with the lambda here, and you can see now that the chiral piece actually also modifies the spectrum, and there's a bit of an active temperature still here, but it all gets modified. So because of this, you can compute your velocity correlation functions again. And you can see you have now still 2 correlation functions, longitudinal and transverse, but they depend both on the rotational diffusion constant and on the chiral amplitude. So I should say chiral amplitude and rotational diffusion are not exactly equivalent in the formalism because they show up somewhat differently in the equations. And so as you can see, here with this value chi in here, and you can compute your spatial correlation functions, and you can compute your time correlation functions, and everything matches each other. So it's a nice solvable system, part of that same class with emergent mesoscopic correlation lengths. If there are no urgent questions right now, I wanted to move on to a slightly different topic, and that's the topic where I'm going to apply this to actual cell sheets. Yes?
When you said mesoscopic correlation length, is there a precise definition?
What I mean by mesoscopic is that long enough that I can use continuum theory in a useful manner, but shorter than system size. Very sort of.
Is it a coarse graining length or a?
No, it appears, it's not a coarse grained length. It appears also microscopically, but like the computation I've done here, they're continuum, right? And so if it was something which was so short range that
That's right,
continuum is
yeah.
inapplicable, I shouldn't be doing this, and I'll show you at the end of this talk that there are cases where one has to be like this. But I say mesoscopic because it's not a diverging length, it will never hit system size.
I see. Okay.
So it's not this is not long range order of any kind.
That's fine, but
Yeah.
it changes, right? So it is a correlation length.
Yeah.
Okay.
Okay, so then let me go on to some cells. So if you want to apply what I just told you to cells more explicitly, we should use better models of cells than simply using soft particles. The soft particles are fun, but we want to do better. So the model I would like to use for applying this formalism to cells is the Self-Propelled-Voronoi model. So the Self-Propelled-Voronoi model, I just realized I don't have the energy. Sorry, this is the curse when you make your talks too late. 1 second, I will stay here for a second, okay? So in the Self-Propelled-Voronoi model, you model every single cell as a polygon. So you approximate this two-dimensional cell sheet by polygonal tiling. And in that polygonal tiling, you associate an energy to every cell. And that energy has a preferred area. So you punish area fluctuations, A minus A0 square. And you punish parameter fluctuations as P minus P0 square. And this was introduced by Max B and co-workers. There should be a reference here, yeah. And what I wanted to say is this has a glass transition in it, or a jamming transition, but the debate is still out. But there is a solid phase in this model as well, which I can melt by driving it in much the same way as I would do with ABPs. So if I take this model, here, and I treat it like active Brownian particles. So these are the same equations of motion as for ABPs, but I'm now taking the gradients of the vertex model potential energy instead of the gradients of like a soft particle potential. Then I can use this in the same manner, and I should say, as I said, there's a melting transition in this model as a function of a shape parameter, P0 over square root of A0, and above driving. So where we want to situate ourselves is somewhere here. So you start in a solid phase, and then if you drive hard enough, you melt. This is also the region where linear response for this model is a reasonable approximation. So I had here, so you can see in that region, you get a nice driven correlated type of motion, which resembles, in fact, quite a lot of the things we saw before, and you can see the mesoscopic correlations in here. Now, in fact, if you repeat that whole analysis, as we did, then you can see here that you see the correlation lengths, you can look at the velocity correlation functions, and you see very much the same as for soft particles, with the exception that you get a different signature of the local positions of the vertices than in soft particles, which is to be expected. And the continuum matches, again, the simulations very well. So, in fact, for all of these models, so the two microscopic models and also the continuum theory, we have something like a master curve of mean square velocity as a function of correlation length. And you can see this as a sort of sliding knob for how in thermal equilibrium am I. So if I'm a correlation length which is 0 in linear response, that's the formal thermal limit. And what I see then here is that my mean square velocity is almost V0. However, the further I go in here, I get into larger persistence regimes with larger length scales. My mean square velocity goes down. In fact, I have elastic energy which is being stored in the deformation. And I can see this as a knob of how far am I from just sitting there in equilibrium, sort of like in linear response, say a sheared system or a system with infinite persistence time would go down here. Of course, beyond linear response, it will get modified. So I'm telling you this because this turned out to be actually a pretty good knob for situating experimental data. So what we did, come on, interesting. We looked at data from my student Kaya Kostanjevec from University of Aberdeen. So these are human corneal epithelial cells which come from the surface of the eye. They're very similar to the kind of MDCK people look at usually. Both are corneal lines. They're slightly different, but not very. And you can see, come on, play, that we can do a PFV analysis on this sort of data set, and you see spatial-temporal correlations in here, which again show the same features which I've shown you before, so space and time correlated. Yeah. The bumps are dividing cells.
Bumps are the dividing
yeah,
cells.
so this is from the surface of the eye. These actually divide quite a bit, so you blink that off normally. So that's you see them coming out of the sheet. I should also say this is quite a long movie, and so you have quite some turnover in the sheet on that time scale. The other the non-white bumps are the nuclei, yes.
The non-white.
Yeah.
And are they confluent or are they dilute?
This is confluent. So there is cells in between here. This is phase contrast. This is not like fluorescently labeled, but there is no gaps in between. I'll show you some other movies of cells in a moment as well. But this is a nice confluent state. Although I should say these are pretty spread out. So they think of fried eggs, yes, in a pan. This is what you're seeing. So if you do analysis now, you can compute velocity correlation function in this data set. And so it's pretty reproducible, and you can fit it with a theory reasonably well with a length scale, which is 5 to 6 cell diameters, or roughly 100 microns. So this is something which is actually reproducible. Many, many epithelial cells show up with that same correlation length over and over again. And if we do this, we can actually fit our theory and situate everything on that graph which I just showed you of the mean square velocity as a function of correlation length. We find ourselves here, and we find ourselves in the highly active regime. So we think this is rather far away from what you'd expect from equilibrium. Then I'm going to skip. We actually managed to fit this quantitatively to the data set, and I'll tell you more about this in a couple of slides for a different system. But with some work, we eventually managed to fit both a particle model, including divisions, and a self-propelled Voronoi model to this system. So they're both, for the correlation functions, good matches to the data set we have. And you say, okay, of course, there's a bunch of parameters, etc., and there are, but not as many as you think. So what's left with is, okay, something like the cell radius or the area, the active velocity, the rotational diffusion constant, and then there isn't much left. So it's the potential stiffness, k over zeta, or the vertex model. These are inverse time scales. So they're quite simple. There are only time scales left in the problem. And then you have another one, which is also time scale, the squeeze out rate, and the ratio of that to division. So you end up with really 2 length scales to time scales in the problem. It's not a lot in the end to fit a data set. So I wanted to show you, okay, one more thing is, so we used the same method then shortly afterwards with a group of Jakob Notbohm, and tried to fit their MDCK data set using the same model. And then what we ended up doing there is actually very reminiscent to also what Ludovic said this morning. We tweaked our alignment model to include a bit of alignment, but sub-threshold, so sub-flocking. And we did this because they have actually very nice experimental data where they can look at the traction forces of the cells with the substrate, and they can see that there is actually a slight correlation between them. So the traction forces are not completely spatially uncorrelated. They have a correlation length of, like, here is one cell of, like, say 20% between neighbors. And this is just what you get if you include just a small amount of correlation in there, just well below the flocking threshold. And that modifies the spatial correlation length, which is longer than the traction correlation length, as you can see here. And we were able to do a nice fit with the same self-propelled Voronoi model. So then I wanted to show you now something new and something unpublished. So my apologies to people who have seen this talk like 15,000 times. What I wanted to show you is work from my student Sander Kamerad in collaboration with the group of Inke Natke and with Jan and with Tani. So we looked at using the same sort of approach to an old problem, and that's the edge. Oh, come on. We'll have to do this manually. So that's finger formation in epithelial cell sheets. My apologies, today is not my computer day. So let me explain what you're seeing here. So you're seeing again, this is an MDCK sheet, a large one now, growing into free space. So there is just substrate. On the other side, this is flat in 2 dimensions, yes? Think of a beach, yeah? And at the bottom is the same sheet analyzed. So red is the edge, and the black arrows is the velocity field using PIV. So what you can see in here is that the way this grows over the edge has this very strong spatiotemporal correlations, which people have called fingers or fingering instability in the literature. And so we're very grateful for Inke because she's managed to give us a giant data set of these. So we could do some very high quality analysis and understand exactly what was going on in here using correlation functions. So a lot of ink has been spilled on this thing, yeah. So everybody has their own favorite model for this, saying it is my favorite instability. For example, through chemical signaling, maybe curvature enhanced motility, maybe leader cells with special properties. So I should say we know they're leader cells, but it's not clear at all that the leader cells are not a result of being in the fingers instead of the cause of the fingers. So they exist, but we're not sure of their mechanical role. Maybe velocity gradients leading to accelerated fronts, maybe pressure-dependent growth rate, and I think I'm missing a couple more of them. So what we would like to test first is something much simpler. So saying, well, we know there's spatially correlated cell motions in there, yes. Now, if I couple this to the edge of this thing, having an actomyosin cable, which we know is there, can these two things together simply get us these finger formations as sort of a driven active polymer style thing of the edge of the system, yes? So we would like to find out about this. So what we're doing is, you've seen this slide before, so we tried to write an active vertex model of the system. So, and what we did differently to what I've shown you already is we actually added one more term, which is pair friction between the cell centers, and this is based on the fact that it should be there, and secondly, and you'll see it, because you can get much better matches to velocity correlation functions explicitly in there. And then the active driving is the same as before, just active Brownian. Now if you put all of this stuff together, and yeah, and map and take it together with modeling the actomyosin cable of the system. So this is interesting. So we can only model the edge if we have an edge. And this is actually very tricky using these vertex models because you need some pretty serious computational geometry. Then, of course, I happen to be lucky and be collaborated with Raskus Knepnek, who is, I would say, one of the world's experts in doing that sort of thing in membranes and similar. And so what we did in the end is we used the Delaunay-Voronoi tiling mapping as inside the AVM, but you managed to close it with a boundary which acts just like a semi-flexible polymer. So it's a boundary which is coupled using the Delaunay triangulation, but it has an energy on it, so it has stretching, it has bending energies. So this is like a worm-like chain stuck to the edge of my system. So, and if you do this, and try to match your experiments, what you find is this. So this is a 50,000 cell simulation. I should say this is a single CPU. It's a good code. It took a while to run, but you can see how it's working. So you can see two things. So you can see the formation of fluctuating things at the edge, yes, and you can see correlations in the interior. The color here, I should say, is area. So that means the red and blue, this is also showing you density fluctuations in the system, which are also correlated. I can play it again. And so you can see what it's doing. Sorry? The huge arrows is because you get T1 transitions, so you get rearrangements in there. And T1 transitions are sometimes non-smooth in vertex models. And so you get intermittently, like large velocities within a rearrangement. It's fine in the simulation. I can tell you the details of exactly what's going on there later, but the simulation is stable across this, yes. Of course, in the theory, we don't handle these. Theory, we stay linear for now, yes. Sorry?
Fluctuations that are coming?
Yeah, so as I said, the large vectors is rearrangements, also called T1 transitions. So so T1 transitions is when you exchange neighbors between cells. I can write it down for you later on. So these are always tricky computationally, but it's well defined in here. Do we have a maybe in the discussion section? Okay, if you can, if somebody has a piece of chalk at some point, yeah. How am I doing in time, actually? Yeah, okay, so T1 is, say, if I have cells, yeah, there's 1, 2, 3, and 4, and these are moving inwards and these are moving outwards. I will eventually go through a 4 vertex and then rearrange in the opposite direction. So this is computationally a complicated process, and we use it through a mapping with a Delaunay, where this is exactly equal to something called an edge flip algorithm. I'm happy to talk about that after I'm done here. So given all of this, we can now try to match this with our theory and understand this and maybe write some more theory. So the first thing we did is we said, okay, let's generalize our active elastic theory to the interior of our system. So it's the same sort of approach, but it's a bit more complex. So as you can see here, this is the active elastic theory for the interior. So you have in continuum bulk deformations, shear deformations, compressive friction, shear friction, and active fluctuations like before. Now you can solve all of this, and I should say the man who did most of this calculation is my postdoc Jan Edwin Keter, so who also was Ludovic's PhD student. And so you work on this, you end up with a Bessel function or a set of Bessel functions with 4 length scales in them. So the first 2, 1 and 3, the bulk and shear active elastic length scales, and you can see still here B plus mu tau over zeta, but you get a friction component here. Same thing for shear, and then you get separately 2 actually hydrodynamic length scales, which correspond to the ratio of bulk friction, and pair friction. Yeah, so substrate friction and pair friction. I should say, in the units we're using, these are all roughly 1 cell diameter. So we have the same amount of friction with the substrate as between particles. And this looks horrible, but at the parameters we're using, this turns out to be essentially indistinguishable from an exponential, okay? So it looks almost exactly like an exponential, and it matches very well with both the simulated and the experimental data that we have. I should say you can also compute time correlation functions. I didn't include them here because it's nasty and you have to do some of it numerically, but you can see experiment and simulation here again are matching very well. They're correlated with roughly 1 or 2 hours, like before, same correlation time scale. And I can tell you about these deviations another time if you'd like to. So we get a good match in space and time correlations. Then once you do this, you can look, in fact, at what happens on my boundary. Yeah. So we can look first at the tangent-tangent correlation functions. And as you can see here, so this is, so as a reminder, a straight line has a tangent-tangent correlation of 1. Yeah, and so and we have something which is still elongated, so we'll always end up with a finite value here. But you can see a length scale emerging here as a function of time in experiment, also in simulation. The same length scale, about 200 micron, also very similar to the length scale in the interior. And I should say something you don't see, however, is an oscillation. So you don't see the thing oscillating, which is what you would expect with an instability. So we think, based on this, doesn't look like an instability, and also, as you can see, it reaches a steady state. Again, not what you'd expect for an instability. Then we looked further. So originally, we were trying to find sorry about this, I have a similar situation with transitions here. We tried to look at the spectra of the height of the interface, but this is numerically complex. There are problems with overhangs, I can tell you if you're interested. So we recently switched to a spatial correlation function. So the height correlation function in space as a function of time, so equal time as a function of space. And so you can see these always have the same thing. They grow as a function of time, and then they stabilize and reach a plateau in the system. Again, no oscillations, no evidence for an instability. Thing is reaching a steady state. So then we said, okay, that looks like we're onto something. Can we maybe understand this computation and understand this more conceptually? So what we did then, and this is probably all I will have time for, we wrote down an active polymer model of the interface. So we said, okay, fine. In linear response, I'm going to say I have a height over the interface. I have a line tension and I have a bending. So these first two terms is exactly over the dynamics for the worm-like chain. And then, okay, I included pair friction because that's what we have already in the system. And then the active driving of the interior. Now, if you do this, as you can see, you get something which looks rather similar to what we see in the experimental simulation. So you see the formation of correlations of things like fingers which have actually a pretty large time scale, much longer than the persistence time of the driving. And so we said, let's analyze this carefully. And Sander did some heroic work here with pen and paper. And we're actually able to match the tangent-tangent correlation functions very well to both experiment and simulation, and also the roughness correlations as well. And very recently, we also found out that we can compute the finger lifetime in this by computing the height time correlation function in this thing. And as you can see here, this is experiment, simulation, and theory now. You get a lifetime of finger which is longer than 10 hours, which is why you could be excused for thinking it's an instability. It's not. It just takes a very, very long time to equilibrate with each other. Do we have any time left? I will, however, take 30 seconds only because I wanted to advertise the last piece, which I don't get time. So it's fluctuating active stresses or long range order in 2 dimensions with Jan. And so the motivation was that, in fact, in active cell sheets, you have stresses which are rather like that and not self-propulsion. And then if you take everybody's favorite model, Ornstein-Uhlenbeck processes, on the tension of junctions and add this onto a vertex model, you get things which are actually fluctuating a lot more than they should be doing. So we looked at this, and we both thought this thing should make mesoscale correlations. The answer is it doesn't. In fact, it's part of a whole class of systems with noise, which has a Laplacian in front of here, where you have action and reaction in the system, and which break fluctuation, dissipation rather severely. Now this stuff, in a nutshell, instead of looking like that for velocity correlation, it's look like that because you get an extra Q square because of the Laplacian in the noise. And so you get the opposite of long-range correlations. You get a damping of the long-range fluctuations. And that gives us bound displacement fluctuations, long-range order in 2 dimensions, and hyperuniformity. And I'm happy to discuss it separately if you're interested. So since I'm out of time, I want to conclude by saying that mesoscopic correlation lengths, they arise in active solids to persistence interacting with elasticity. They're very generic, and they show up in the system I showed you, and I'm certain many other ones. I would like to acknowledge everyone who was involved in here, so especially Sander, Jan, Zichen, Kaya, the students involved in this, and Rasko and Eric for theory and simulations. Thank you very much.
So while Shiva sets up his thing, we have a time for one quick question. If there's, yeah.
Okay, so in your polymer model, VF was a colored noise, right?
Yes.
So then this model is actually very close to one by Pro and Bruinsma from 1990s of membrane with pumps.
I'd like to talk about this, and I think Tani is saying something similar, so we still need to see how. I think it's related. What's a bit tricky here is we're not calculating nothing which is in steady state. So it's really tricky with the correlations you need to compute. And so it's hard to compare it all exactly to these existing models.
So if there are no further questions, thanks Ilke for the interesting talk.
Would you like your
Yeah, thank you. Starting from now on.
So next talk is going to be by Shiva, and he's going to talk about bacterial turbulence broadly. Shiva, go ahead.
Great. Thank you, everyone. Let me start by thanking Arnav and Pinaki for giving this wonderful opportunity to discuss some of our results. And I am Shiva from IIT Kanpur. So before I'm going into the topic of discussion today, what I'll possibly do is try to change the slide first. Doesn't look like working. Okay, great. So yeah, so I'll take one slide to introduce broadly what do we do in our lab. So I'm an experimentalist, and we broadly focus on two aspects in our lab. One of the aspect is polymers effectively with a specification towards non-equilibrium conditions. We kind of prepare the system, drive the system to out of equilibrium situations. We quench them, and then we try and study them towards their towards equilibrium and stuff like that. And by doing that, our focus effectively is in controlling structure formations and different kinds of instabilities. And also, once we quench it, one ways of getting that idea is to kind of get focus on residual stresses and stuff like that. What we are trying to look at is the molecular reasoning underlying this kind of residual stresses, which is one aspect that we work on. And the another aspect is also we kind of think on understanding the dynamics of bacterial communities, both at large density bacterial suspensions, trying to understand their collective dynamics, looking at aspects of turbulence, and where we recently have shown that in contrast to conventional turbulence, bacterial turbulence do show very strong presence of Levi walk until the fluid is correlated and stuff like that. And also we kind of do a very close collaboration with biologists, where we kind of try to understand how stresses play a significant role in switching the lifestyles of bacteria and stuff like that, right? So with that, which is not what I'm going to discuss today, and what we'll be discussing today is some recent aspects which is not yet published, which we are summarizing the results, trying to understand simple deposition of particles, and instead of depositing passive particles, we are replacing the passive particles with those particles which can now swim and come and attach to the surface, and may not absorb with the full probability, may kind of diffuse through and then get absorbed kind of a little longer. So by just introducing this degree of freedom, so how does results change, and the kind of surprises that what we have seen. And of course it's important that I highlight that this is from like a lot of hard work from these two graduate students from my group. This project is led by Hemlata, and Dhananjay is also sitting here in the audience. And as I said, like, we do a lot of collaboration with these biology guys, and like, we get the bacteria from them, and that's Saravan from biology department, and thanks to all the funders for funding our activities. And yeah, so let's start. Like, when I think about interfaces, like, growing interfaces is something that, we see in various aspects, right? Like on left, what I show you here is like a Belousov-Zabardonsky reaction, like, chemical oscillations which are growing, and then, like, on the right, you might have seen the movie, like, these are turbulent liquid crystals where the different phases kind of, like, grow into the other phases. And people have seen, like, ink imbibition and paper combustion and stuff like that. Interfaces which are growing is something that we all have seen in one of the other forms. And also in the living systems, there are a lot of very interesting aspects. Like on left, what I show you here is a dendritic swarming colony of a bacteria, which is an interface which is growing, and this is at a much larger scale. At the local scale, the bacteria is still swarming, and, like, you see a lot of collective dynamics, and which drives such patterns. And also, like, people have shown with monkey cells, like, on left, what I show you is monkey cells where there's interfaces which are growing and so on, right? And just to give some background, especially for the younger colleagues here, to give some aspect of how do we think about deposition of particles, right? So just think about, like, particles which are coming and settling down, and, like, if one wants to write a simple continuum equation with respect to that, so we are thinking about an height which is growing in time and which is kind of getting smoothened like a surface tension like term that you are having, and then you have a stochastic noise in here, and then this is an equation which one can analytically solve. And, like, if one does this for a one-dimensional system with another one being time, like, one gets the roughness of this interface scaling with the system's size to the power of, and the roughness with time to the power 1/4. Of course, this is at much later times when the system has kind of reached saturation and stuff like that. And then another thing which the famous Kardar-Parisi-Zhang's inclusion, which is, like, if one thinks locally, there are, like, nonlinear corrections one has to think about for the flux field, and if one does that, like, that changes the exponents, both of this roughness and the growth exponents, right? And just to summarize, like, effectively what we are thinking about are interfaces which often follow power laws, like, where the roughness scales with the size of the system, or in the Fourier space it scales with the wave vector, with is also, again, one can get the roughness exponent in both the ways, or one can think about the time evolution of the roughness, or, like, if since the flux is linear, I mean constant, one can also, like, replace time with the mean height of the interface and stuff like that people have been showing in different experiments, right? And then thanks to Vichak family scaling, one also now knows that, like, the relation between alpha and beta, while the time correlations like come in at smaller times, at later times when you see a saturation with time, then the length scale correlations becomes much more evident, and the relation between alpha and beta are captured by what is called as this dynamic exponent. Is that right? So that's all about the background. And given this background, what we thought about this, we have discussed about the ideas of this, and, like, there are papers which are published which is saying that, like, one can essentially experimentally capture this idea and one can map it on to KPC type of interfaces and stuff like that. And what if I have now an interface which is formed, and now as you can see that, like, it's not just that, like, it is kind of advected towards the edge only, like, that they can now also, like, do some random motion. If I think about an interface which is forming like this and that it is growing, what kind of surprises or what kind of aspect that one can think about, right? And the focus of today's talk is trying to understand, set up this kind of interface, and then look at the results both from the experimental perspective and a very simplified description of model, right? Okay, so yeah, first we want to create interfaces, and creating interfaces is not a straightforward task, and we just used a very simple idea. We are all used to stain formation of a coffee drop when it falls on our shirts, on our tables and stuff like that. And if you see the coffee drop, you see the edge is, like, much more thicker which is like the coffee particles are much larger in terms of concentration at the edge. And why is this happening? If one thinks about it, just put in a drop, and then because of the geometry, the evaporation at the edge is going to be larger, and now just to accommodate space, you would lose lot of volume here, but then this is energetically much more costly, which means to have surface energy minimization, one has to have an advection of the bulk fluid from the center of the drop towards the edge of the drop. Now assume the same thing with particles. Now if you have the particles, while the central fluid is getting advected, it also drags the particles towards the edges. So now if I sit on here and then look from the top surface, I am seeing an interface which is being formed. Right? So that's the idea that we are going to look into. And then, as we promised, the first thing is we want to compare the idea with active system and a passive system. So for that, what we chose, of course, geometry is important. So I cannot just replace living system with some other shaped static passive particles. So that's why we chose two different bacterial systems. One is Bacillus subtilis, which is like a very strongly swimming entity in our case, and E. coli, whose swimming is strongly repressed. Right, and now what I'm showing you here is the trajectories which are averaged over I think few seconds, and the color maps indicate the velocities, and here I'm showing you the distribution of the velocities, you can speed effectively, and you can see that the guys which are swimming effectively have a swimming velocity of the order of 20-30 micrometer per second, and the guys which are not swimming are kind of most localized, and if you look at the velocities, this is more than an order of magnitude smaller. So effectively one can think about an interface which is forming with Bacillus and an interface which is forming with E. coli, and try to find out what kind of interfaces we have. Right, so that's a videos I think a little accelerated, 8 times faster than the real timings of the experiments. You can see, let me rerun the video. Right, so now the passive guys more or less get advected because of this advection of the fluid flow, while the swimmers, you can see they are coming and forming this interface. There's a finite interface which is forming, but it is not that it is kind of coming and just attaching. They also can move around, right? And now if one thinks about again the velocities of the advection and the swimming, what is interesting is that even though these guys are not moving much, the advection flow is much stronger, that the velocity to which they are coming and forming the interface, the flux is very similar in both the cases, so that there is no difference between the active and passive systems in terms of the flux, right? And with that, now we have to characterize the interface, and now what I'm showing you here is fast forwarded video with the red line which is effectively capturing the interface. The one bacteria comes and falling, right? And one focuses it, as Silka has also highlighted, that one can see certainly overhangs which are visible, and hence defining H as a single valued function becomes problematic. But then there are earlier works including a review by Takeuchi, where one can show that if the overhangs are not very strong, one can in principle neglect that and define whatever the maximum height that one has as the single valued height function. And while not just kind of inspired by the literature, we also thought okay, let's just do our own system. Now what I'm showing you here is that the actual interface which is shown in the black squares, and now the red lines are effectively taking the height at every location, whatever the maximum height is. So I'm neglecting overhangs in one case and including overhangs in another case, right? And then we did just a simple box counting method and try to find out what the dimensions of this fractal object what we are having, and one can see that they're coming out to be pretty closer, and we have done it for different times and different species, and it's not changing much, and hence neglecting overhangs is not a very serious problem in our case, I presume, and that's how we go ahead. So I define height as the maximum at a given location, and as a function of time this evolves, and then to support this analysis, I also parallelly show the factor analysis where I don't neglect the overhangs, right? So that's essentially the background. So now for that, of course, I define the basic statistical parameters. One gets h at every x location as a function of time, and you define the mean height and the roughness around the mean height, and that gives you the two statistical characteristics that one looks into. As I started introducing, what we are interested in is in correlating evolution of roughness with time. time, or in other words, correlating roughness with mean height. In experiments, finding zero time is little difficult, and that's why we usually rely on correlating roughness and mean height, right? Okay, so that's what I'm showing you here. The first results, and this you have seen already. And now if I plot the roughness as a function of mean height, phi m defines the fraction of the swimmers. So phi m0 is basically a passive particle, passive bacteria E. coli, and phi m equal to 1 is like all of them are swimming, is a Bacillus subtilis. And you can see, like, by looking at this evolution, the exponents for the swimmer and the non-swimmers are strongly different, right? And just to give you some, so this ash color symbols, what you are having is the actual experimental data, which is collected like every 30 frames per second. And then this on top, what you are seeing is blue symbols are every 3 seconds I'm putting in one symbol, just to show like that, it's not much is varying in terms of the time there. Right. So then if really wanted to identify if such transition really exists, what would be like ideal is to one think about systems which has different activities, or one think about populations which has different populations of active system in a suspension, right? So how do we do that? We cannot just mix E. coli and Bacillus, because biologically they create other issues, and that's why what we decided is like, okay, this guy is swimming really fast, and let's just now remove the nutrients from them and controllably remove the nutrients, so that you are playing with the metabolic activities of the system. So you keep waiting, so then slowly they start losing their activity, right? And that's what I show you here. Sorry? Yes.
So that's not 1/3?
It's not 1/3. For instance, experiment similar to this was performed in Arjun Yacht's group with rod-like particles. No, rod-like particles show kind of quench KPC. Okay. So they show that like the beta is of the order of 0.67, 0.7, and their alpha is also comparably different. So it's not KPC class. Right. Yeah. Okay, so now what I'm showing you here is I took the swimmers, removed the nutrient at time t equal to 0. This is how they are swimming, and I just waited for a few hours, and you can see while the density, the number of swimmers are very similar, so one can still think that the particle number is conserved throughout this, and they are not multiplying because of the reduced metabolic activity. And one can see that here all of them are swimming, here few of them are swimming, and few of them are more or less static, as though you kind of separate a population of guys which are swimming and guys which are not swimming. So then we looked into the distribution of this speed, and if you just see t equal to 0 minute, you can see this long tail which is coming, and with a peak at lower value, and with time you can see the peak at the lower value keeps increasing, and the tail keeps shrinking. So effectively the population which is swimming is kind of decreasing, and the population which is non-swimming is increasing. So now based on this idea, I can effectively integrate out the area of this regions, and I can define a fraction, motile fraction, as the ratio of these two areas, right? And that's how we defined the motile fraction. And once we define and then get it for all the systems that we have done, and you can see that this is our pure swimmer. Now we can go from something like 0.7 fraction to something like 0.1, and then 0, which we had this non-swimmer, two extreme cases, and that now we can control in between, right? And now let me show the first results with this mixed population. You can see phi m 0.68, which is 68% of them are swimmers, and the remaining are non-swimmers, and down to phi m 0.1, which is 90% of them are non-swimmers and 10% is swimmers. And you can see while the guys which are swimmers display very similar exponent as what we have seen for the pure active system, the ones in between, the mixed population, almost 50% of mixtures of swimmers and non-swimmer show a lesser exponent, which means that the kind of roughening is a smoothing is more efficient at an optimal fraction in between, right? And just we'll go ahead and show you little more results on that. We just wanted to also look at other characteristic exponents. For instance, we were looking at the roughness exponents, and none of this activity seems to really change the exponent here, and which indicates alpha is of the order of 0.45, and within the exponent one can say it's 0.5 or something like that. And then we also did this, without neglecting the overhangs, looked at the fractal dimension calculation. The fractal dimension seems constant, and we have done for a series of systems here. And now beta, which is the growth exponent, is now plotted as a function of phi m. Alpha and df is also plotted as a function of phi m. Both alpha and d are not changing with phi m, which is more or less constant. And the surprise comes when beta seems to have a non-monotonous behavior with phi m. For purely active systems, completely swimmers, we had something like 0.5, and in between it kind of goes via this 0.3-ish. I don't know if I can call this as KPC, but I'm refraining myself from not calling it with anything. So this is just some decrease in the growth exponent values and then increases again. Yes, Satya. I don't think so. I don't think Wiechert scaling will hold, because now your alpha and beta are supposed to be coupled. Now we see that alpha and beta are decoupled, right?
It could be that Z itself changes.
It could be. So that's why, if one just takes a ratio, it would indicate that is changing. But I don't know if I would rely on family Wiechert kind of scaling to rescale things and stuff like that. Right? Yeah. So that's one. Such decoupling between beta and alpha is not predicted, and also this fractal dimension being constant is something one would have expected with alpha being constant, because you know that alpha and the fractal dimension are related to each other. And so we started asking, okay, these are all at a interface level, and we don't know why this is happening. So maybe we will find some clues of looking at the process of how this interface is forming, right? So let me take you through what we think could possibly happen. Right? So now we started with a sessile drop, right? And now we are thinking about an object which is floating on a sessile drop. And now any object which is floating on a fluid, you know that it creates this capillary deformation. And now let's say if this object is moving, right? And now if this movement is more faster than the relaxation of the capillary deformation that what we are having, effectively what you have created is a trail of capillary deformation. And now if there are some other bacteria which are coming here and here, so they would rather be attracted towards this trail because of the Laplace pressure, right? So now if that is happening, maybe that could indicate why we have this very rough beta being 0.7 or something is happening. Just we looked into experimental clues if this is happening. Now if I just kind of bring your attention towards some of this trajectories which they are coming and forming the interface, you can see that they kind of switch lanes and kind of coming and forming at a given location more prominently. This indicates that somehow they are going towards a given spatial location, which might be the reason behind the larger beta what we are seeing. And then now this is for a passive system. Here activity is not there. This is just the geometry which is playing a role. So now the moment I put in activity, now if you see this swimming guys are moving around the interface, which means if such quenching lines are existing, so then they would rather randomize the line, and then one would expect that a randomized value is what one would see, and beta being 0.5 seem to indicate that. But what is the reason why we have an optimal fraction in between where the beta is much smaller, that we don't know yet. And now if this smoothing is what is happening, so what we did is we kind of looked into the height fluctuations, right? So what I'm plotting here is the mean height fluctuation, which is ensemble averaged for different phi m's as indicated here. And now our interface is always growing in time, so I have to appropriately normalize with the growth of the mean height, and we are removing that. We are now looking at only the fluctuations. And now if the active particles are coming and smoothing this interface, what would you expect is that this fluctuation is now not happening as strong as without activity, right? And that's what this gamma is indicating. Now with activity, with increase in phi m, you can see that the slope is decreasing systematically, which says that all that the guys are doing at a given location is effectively effectively it is trying to smoothen the fluctuations. Now this is at the height level. Now if you think about the particles are now also moving around, right? And now if I look at the mean square displacement of these particles which are right at the interface, now at delta t equal to 0, the particle has just come and stayed as a part of the interface, and then now we are tracking this particle, right? These particles now, if I have an active system, they are like kind of really swimming around, and that exponent indicates that you're really having a like kind of a very almost a ballistic kind of motion. And now if I have like non-swimmers, like passive guys, they are coming, they just rearrange locally, close to being random, and then just get settled completely, right? And now if I think about the mixed population, right, and there we have seen all type of entities. So there are entities which are swimming around because we have a population which is swimming, and then there is a population which is not swimming, and then there is also another population which is kind of trying to swim around, but then get stuck because there is a region which is growing. They are trying to swim around here, and this is going faster, and then this is just getting stuck, as though you are feeding this growing interface from the sides, right? And that is something that what we have seen. And also while we are looking at this, another thing which came to our attention is that when you look at the interface which is forming, so this is at time t equal to 1, this is with increasing time, a bacteria which is coming and becoming a part of the interface here. So now with time what happens is they kind of jump around and then finally get settled at a different location, right? So now this is what I call as this length, the jump length, before they become a part of this interface. And now this jump length, we decided to just look at the distribution of this jump length for different systems, pure swimmers to passive guys. And now the first one, let's go to the passive guys. What is happening is now I'm showing you a rescaled jump length with the length of the bacteria, and you can see all the jump, there is no jump beyond the length scale of the bacteria itself. So they come, they just rearrange, and then they just get stuck. That's what is happening with the passive system. And for the active system, if you now focus on this black square symbols, you can see that more or less for almost up to 10 times the bacterial length, all of them kind of move around before they can get stuck, right? And then it starts decaying much more strongly, and in mixed populations we have a slope which is different from the passive population. So it has more larger jump length distribution, right? So this we will come back to little later. Before I'm going further, let me just also think why we are seeing alpha constant, right? So now the way we are doing is this is the actual interface, and now when I recast into this height variation, which is plotted normalized with the mean height. Now this video is a time video. Now you can see that there are movements across this interface, which is kind of reflecting the movement of the bacteria along the interface. And how do we calculate the alpha? We are calculating alpha at the latest stage, so hence swimming is not going to really matter. Even if that happens, we also looked into different times. Now we are kind of going to get different scales. At a small scale you get W from each box and average it out, and at large scale again you average out. So whether the bacteria is being a part of this box or this box, it actually doesn't matter, because it is kind of getting averaged out finally, and that's why we think alpha is not changing, right? So now just to summarize, the main thing which seems to happen is that that before the interface is forming, there is a new degree of freedom which was not discussed in earlier deposition of particles, which is effectively you have this jump length distribution. So if one tries to summarize the idea of what experiments indicate, so basically the non-motile guys come in and get absorbed with a probability 1 and with no jump length probability, and the motile guys coming and more or less they are not absorbing at all the moment they are seeing, they move around and then they get absorbed for a finite length distribution, and the mixture will have a contribution from both of these guys, right? And if this is really kind of summarizing the ideas that we were discussing, so then in principle I should be able to set up very simplified model, just trying to bring in these ideas into a well-known model, right? So that's what we tried doing. So to start with, we chose like a ballistic deposition model, and where we chose in such a way that there are spurge of particles which are coming and settling, like 800 particles at a given time. which means they can now get attached in whichever phase they are seeing. So we just allowed the overhangs because to mimic similarly what happening in experiments. And so these are sticky particles, so as I said, like they can get attached wherever they are seeing the first neighbor. And then, like, you know, we are using a box of length 1000 with 1 being the particle size. And so at a given time we are sending in 800 particles, and we are waiting for 10,000 steps and seeing like the interface is going, right. So now this is a nominal system, and now we have to bring in the experimental inspirations into this model. So what did we do to think about this geometry induced quenching? Like there are some regions where they can, like, you know, particularly go and get settled. To mimic that, what we did is, like, you know, when these particles are coming, they also have an extra additional freedom. They can move around for 7 pixels, and if they find any neighbor in any of the 7 pixels, they first get attached there, which means if you have some regions which are growing, they will grow at the expense of the other regions, right. So this so then if that is growing, so then like you are having now few locations which are growing more prominently than the other regions. So that kind of try to mimic the quenching that what we are seeing in the experiments. And then we have to also bring in the activity. So the way we did is that, like, you know, now the particles are coming, so now we are for each particle we are giving in a probability on top of that, and this probability is, like, you know, now Pm minus 1 is the probability with which they come just attach, and Pm is same as phi m what we discussed in the experiment. So then they can, like, you know, if you now control this population difference, they can get attached or they can move and stuff like that. So that's the idea behind this model. And with that, now let's first show you the flux that we got. So all the flux is more or less linear for different systems, and now we wanted to do exactly same things what we did in the experiments. Look for the maximum height, look for the roughness, and look for the correlation between the parameters, and that's what I'm showing you here for all the systems. Right. So let me take you through different graphs. So this is a non-motile guy. So now what we said, like d equal to 7. So they can move around. There is a particular location which can grow. So which means your beta is expected to be larger, and we see beta is close to 0.6, 0.67, and then it saturates at a larger region, and which is expected, Raja-Lin scale. Right. And then you know, for the motile guy, so they are coming and then they kind of randomizing everything. So there is that probability we just brought in with jump. The probability of jump is 1. They can now move around, like from ranging from 20 to 1000 you know length, and the probability of all of them is equal. Right. So now with that we could get similar to what we were getting from the experiment, which is 0.5. And then for the mixed population, what we see is, like, initially we still had something similar to 0.66, because the passive guys are, like, you know, kind of like, you know, kind of dominating at the initial time scales or the initial mean heights, and then later on, like, you know, kind of saturates towards getting into 0.33, right. And then, like, you know, this is just like what I summarized. So now betas on this dashed line is what I just shown you in experiment. So effectively this simplified description seems to capture that the variation in beta is coming from the phi m fraction what we had. And then of course we have to also look into whether the alpha is being constant or not, and we looked into the PSD, and that seems that, like, you know, it's more or less constant for the simulations what we have seen. Right. So with that I come to the conclusion, and I hope I conveyed the excitement of our experiments, like, you know, where controlling the swimmer fraction in experiments and in the simplified model that what we are showing you know, allows us to switch the growth exponents from, like, you know, 0.67 to 0.3 and to 0.5 in a very non-monotonous manner, and such decoupling of growth and roughness exponents are not predictor or expected, and hence I think there is something more to think and discuss about. So with that, thank you.
Thanks, sure. Time.
Yeah. So, I mean, in your experiment, I mean, you don't look at really the finite size effect, right? I mean, the family.
The drop size is really larger.
But you could look at the spatial height correlation function. That's another way of getting the Z and alpha and beta things, right? Did you look at that?
We looked into not really a correlation function, but I thought, like, you know, PSD is somehow reflecting that.
No. I mean, you know, if you take two spatial points.
Correct.
And you look at the height correlation
okay.
function in the growing region. So imagine that your system size is very large, and you're just two specially separated points and look at the height-height correlation function as a function of x and t.
I see.
And again, you will see the similar family Wittig scaling function. So that's another way of getting the exponent alpha and beta both from the growing regime instead of waiting for it to saturate. Fine.
I see. So we'll look into it. Yeah. Thanks.
And the other second thing is that you could also look at the height distribution in the growing regime.
Mm.
That is, if I look at the distribution of height at a given point in space. So this is just the first two moments. Width is
Yes. So we have.
the full height distribution is known to be, for the KPC equation, it's given by this Tracy-Widom distribution.
Correct. So we kind of looked into the skewness. The data fluctuation is larger, but if one kind of looks at the values, it seems the passive guy seems to converge towards what is expected from Tracy-Widom distribution. The active guy seems to show much larger skewness. So I'll have to look more.
So I guess, I mean, I don't know if I understood what you were saying, but I mean, could these, the fact that you're seeing these exponents changing, could it be sort of an artifact of where, in what regime you are? I mean, if there are crossovers, you
Right.
know, for the different phi m's, could you be in different regimes where you're estimating?
Well, so the regime transitions usually is like, there is a regime where your roughness scales, like, there is a kinetic roughening, and then, like, it kind of saturates, and then only the other regime really takes over, where your alpha seems to contribute. So in our case, what we have seen is that, like, our roughness is always increasing with the times, with the experimental window what we are seeing. So I think we are always in the same regime, and whatever transitions we are seeing is beyond the regime switches.
Yeah, the presence of overhangs would be much more in the passive case compared to the active case, right? So that could also affect your exponents because they're not very different exponents.
Yeah.
Right?
So we kind of did the same reference with all the fractions that we have worked with. Overhangs, with and without overhangs, we have not seen much variation in terms of the fractal dimension or in terms of the roughness exponent. Right. So now for any time evolution, one has to think about some way of capturing this time evolution. That's why we kind of went ahead with these neglecting the surface, the overhangs. Right. I'm not sure if they are having any differences in terms of phi m. So we have looked into and we don't see any difference in terms of the extent of overhangs.
Yes. Question, comment in the same direction. So for us, we had trouble with overhangs. And for example, we had naively tried to compute spectra in our system. We're very happy because they look good, and then realized most of our spectra was overhang noise. So did you have any problems like that?
So when you say
that would be quite tricky, yes.
When you say spectra, you mean in spatial?
Fourier transform of our height field in the end.
Yeah, that's what I was showing in terms of neglecting overhangs and remove, like with this box counting method. So this being similar, I'm not sure if that is going to be a real reason behind any of our observations.
For us, it turned to be much worse than we thought it would be because we have very few actually, but yeah.
No, I agree with you because like that's what we also thought when we started with. But then when we did this analysis, we could clearly see that overhangs are not really playing a dominant role.
All right, so if there are no further questions, let's thank Shiva for an interesting talk. We break for coffee and there is an announcement by Pinaki.
Yeah, a couple of announcements. First announcement is we come back at 4:30. So unfortunately, Ariel couldn't make it because he got injured, so he will be hooked up online. So he joins at 1 p.m. Israel time, and that's 4:30 for us. And the other announcement is for the students. So for the conference dinner, we will meet at the gate at 6:30.